is a technology and a concept. I think we have been always, we talk about phenomena related to water. We have lots of experiments. We prove repeatability results. But I've been asking many times why we did get these results. And by asking myself why, gradually, so we started with the diagnosis of disease using light, and meanwhile I'll explain what kind of light. And by trying to explain why we see this possibility to diagnose disease, we ended up understanding that this is because we see different water spectral patterns. Now, what these water spectral patterns mean is something that I'll talk about today. Now, I'll talk about, first I'll introduce to you Aquaphotomics. Is a concept that we introduced. It means interaction of water and light. Well, these are the two characters that mean that it's very easy to write and beautifully calligraphy. So it's all about water and light interaction at various frequencies of electromagnetic spectrum. The idea is that water, because it's a system is a molecular system that interacts with the rest of the molecules in the system, water works as a mirror. As a mirror when illuminated with infrared light. Because visible light is reflected by water, just a little bit of light. Infrared light is absorbed but not fully absorbed by infrared. And so infrared light goes into the water, gets absorbed at certain level and gets back. So everybody knows, we talk about information, we talk about water memory, we talk about coherence. The light that comes back out of the system after the light has been interacting with water might have some information, I think. Okay, we're not that. Yes. So, water is a molecular mirror. Infrared light, where is infrared light? Right next to the visible light. A lower energy than the visible light, but not absorbed as fully as infrared. Well, this is just basic knowledge where infrared is next to the visible and infrared. And infrared also, the light could be transmitted, absorbed, reflected. Infrared is absorbed and could penetrate to a very low distance. Now, this is something very interesting. This is the intensity of light coming from the sun. And what is interesting is that this part is the UV light, this is the visible light, and from here to the end, half of the light of the sun is in the infrared region. So we still don't know why. What is the purpose of this infrared light? And remember this peak, this is the intensity. High intensity of sunlight in the visible light, in the visible range. Lower absorbance of water, this is how water absorbs light over the electromagnetic spectrum. Water absorbs lots of UV light, lots of infrared light, but very low, little of infrared and terahertz and sound. Okay? So what we use this infrared just in the middle to get information from the water molecular system. The whole concept is water is very sensitive, water gets perturbed by any and many different perturbations. Very small influence could change the water the arrangement of water molecules. Now we can take a spectra. This is the water spectra. If you look at it with your naked eyes, you don't see anything. And the changes usually are in absorbance units are 10 to minus 4, which is very small. If we don't go up the ground, we don't see it. If we don't analyze with numbers, we don't see it. So we need to analyze multivariately those data. Now, we talk about water matrix coordinates. I call them coordinates because certain water absorbance bands, they play as hubs. Hub means they get information or they give information. So if we look at those windows, those coordinates in different combinations and different amplitude make a spectral pattern that we can use it as a biomarker for diagnosis. So we have this, these are, for example, 12 specific water absorbance bands that we found in the first overtone of the water. So we could say, if we look at these 12 bands, we can harvest information for the water molecular system, which reflects the molecules in the middle, the rest of the molecules. We could have one single molecule, and that single molecule at a certain time will change the water molecular system in a unique way. So if we know this spectral pattern, then we can use it to identify this single molecule. Aquaphotomics, like proteome and genome, is a database of all specific water absorbance bands and water changes different systems. It's the entire complement of water matrix coordinates. Now, one example, so we are here different systems like parent protein, milk protein, blood, urine, in different samples. When we perturb those samples with different concentrations of temperature, we could see different spectra of different water. These are the water bands, 12 water bands. These were activated when this is healthy milk from healthy cows. When the cow is healthy, when it is sick, they get changed. So we use them as the coordinates of the biomarker for healthy and unhealthy diagnosis. And we do assignment. So assignment means that we understand specific band, like this is the band which is the third of the antisymmetric stretching, and this is the water solvation shell, 30 and 60 nanometers to this region. And it comes from the many papers and recently more and more. People in infrared, they make an artificial system and they identify those bands. So we calculate the first overtone and we find a very nice agreement and consistency with the bands that we found in a multivariate way as bands that show higher variation in the spectra. So we first look at the bands with the high variation and then we try to identify those bands, and we find agreement with already identified bands in the infrared region, calculate the first overtone, and this is what we get. I showed just 4 to explain to you. Aquagram is a graph. Just to see the difference in the spectral pattern. Aquagram is based on these 12 bands, and this is the spectrum of water vapor and liquid, just water. And this is how we see these 2 spectra at these 12 specific bands. So you cannot mix them. I mean, you cannot mistake. You can see that the water vapor absorbs high energy here, and one of those bands is a water solvation shell, specific structure, which is very interesting. But we don't have it we have a little bit, only a little bit in the water We do have vapor in the water, and we do have ice in the water, but the concentration is very low. Now, just to show you an application, because we had very interesting talks, and this is the moon cycle, so we have followed different waters over 3 months, and how the moon phase influenced the water. So I will show you, these are the aquagrams of these different kinds of waters. Just we put different names, and you see the color means different water, and this is 0, 0 day, 3rd, 6th, 9, 15, 18, 21, 24. Exactly, this is related to the talk about the trees. And you could see that we have specific pattern, spectral pattern changes. You see this violet flipping to the, these are less hydrogen bonded structures, and these are all weak hydrogen bonded structures, and these are hydrogen bonded. So we have these two, and in between, of course, also changes of different coordinates. So just to show you an example. This is what we can do with this concept. We can, having this WOMAX and Aquaphotom, and the information from the spectra, we can take a holistic approach, a system approach, which directly goes to functionality. This is the point. So we don't want to go molecular by molecular. We want to go by a spectral pattern, a water spectral pattern, that mirrors a whole bunch of molecules that are in the system, and is directly related to the functionality of the whole system. That's the point. So then we could have lots of applications, but before that, probably the point that I like is that we can understand what is happening. What is the structure that is changing? What, for example, I've been trying, and I still feel a little bit guilty toward Jerry, because I haven't done a presentation about the whole spectral pattern of easy zone, because easy zone is a spectral pattern. It is not one single coordinate. It is not, there are not two coordinates, and they're different systems with different spectral pattern. So it will come, I'm sure. Now, ion hydration, we just got, our paper got accepted after 1 year rejection, so I'm very happy to tell you about, this is a work together with the Keio University in Tokyo, and this is about ion hydration. Why it got rejected so many times, and for 1 year? Because people think that they know everything about hydration, about ion hydration. They just didn't want to look at the paper. The point is that our point was we started with very low concentration. This is the range. And we tried these halogens and metals. So we wanted to show what kind of spectral pattern we obtain, and what is the specific pattern. And this is the situation, well, we have a plus and minus, and we found this specific dehydration band, we call it dehydration band, and hydration band. You could see the differences. There are differences in the strength on the covalent bond specific for the water that is captured between the ions and for the water that hydrates the ions. So we can see it in the spectra, and I'll show you. This is just the spectra, first overtone of the water for different ions. You see, this is the pure water. It shifts towards high energy absorbance. It makes a blue shift in general. Now, when we subtract the control, what we see is this is the dehydration band. This is the water that decreases when we dissolve, when we lower the concentration. The water that is stuck between these two ions decreases because the ions are separated, and we lose this type of water. We lose this strength that we can measure at 3092, 3098 nanometers. And on the other side, we have rays of these bands which have a very strong shift. This is the dimer, this is the water dimer,
but this is up to the water dimer. It starts from 1412, which is the free water molecules, cold free water molecules. We still don't know what it is. And it goes to the water dimer. Now, so we call this hydration band, and we call this dehydration. And this is the spectral pattern, which is typical for ion hydration. Now, we did an experiment, which is similar to this experiment, just to prove that whatever we do the experiment, we analyze spectra in Ireland and Rome and Kobe University. We had these 3 laboratories doing absolutely the same experiment. This is the experiment. We had these salts at these concentrations, and we wanted to prove that we will always get the same results even though we have that low concentration, and infrared spectroscopy can, those metals and salt, they do not have absorbance bands in the infrared range. So everything we're looking at those is water, okay? And look, you could see that we have these salts with absolutely the same spectral pattern. So this is we published in Talanta, and this proves, this is very much related to what you just saw. This is the dehydration band, this is the hydration band, and this is the hydrogen bonded water that is decreasing with ion hydration. So we can explain, we can understand, we can see what is happening with ion hydration. And the beauty of the infrared is that we can have that small, half of my small instrument, that we can bring it, we can use it for real monitoring of water, for diagnosis, etc. And this is the model that we did, and we proved that we can measure this low concentration of ions. And the lowest error is exactly in the first overtone of water. Not more, not less. The first overtone of water, where we use all the water-specific bands, if we use them, we get the lowest error in our model to measure low concentration of salts. Now, I will show you now how we can use this basic, because what we're doing, we try to understand what's happening, to have a clear picture of, with numbers, and how we can use it. The first is we got a paper accepted in +1 on the fibrillation of insulin. And this is the concentration we're talking about. We took the spectra in the whole infrared range, but we show that if we use only the first overtone, only the water information, not the protein, the protein concentration is very low, but if we use the water structure, this is what we get. Our question was, we wanted to know what is happening in the nucleation phase, what the water is doing, how the water is interacting with the protein, what is the environment that the water creates, and as a result, we get this nucleation. Now, this is the first seconds and minutes measurement, the first few measurements, when the temperature is here. So we could see that we have dehydration, this is the dehydration band, and hydration. So you could see a very prominent spectral pattern related to dehydration and hydration. Now, this is when we have less, the hydrogen bonded structures increase. This is the next step in the process. This is a reaction that we monitor over the time. We have a cuvette cell with the protein in the instrument, and we just continue, do continuous measurement of the spectra within an hour. And this is the final stage when we have elongation, and we already have a hydration of this elongated part, and again, the hydration band at 1425. So, the next example I'm going to give is about the oxidative stress diagnosis. So I don't think I have more time, but the The oxidative stress, I just want to show you how the spectra, this is the first overtone, 1300 nanometer to 16. And the blue are cells that are oxidized. So we monitored cells over a few hours to see what is the difference between the healthy cells and the oxidized cells. And this is subtracted, the first spectrum is subtracted, and we could see then, very interestingly, in the oxidized cells, we lose this working part. 1464 nanometers is a specific water band related to proteins, to interaction between water and protein. It is called working horses. There is a paper on in infrared spectroscopy. So we got consistency. We know that this region is related to, it's called working horses. Now, we did an experiment with mouse. And after using different feed, adding to additives to like this to get pro-oxidants. So we monitored for a few hours, and we got this spectra. This is the short wavelength region, which is easy to be used for the tissue measurement. These are tissue measurement, and also measurement of the blood and the organs tissue. Now, this is abdomen T-bars measurement, a model to measure T-bars based on the spectra of rat abdomen. So as you could see, we got very nice correlation between the spectra and the T-bars. So which means the spectra related to the oxidation. And another example is the rat liver. So we got this, but you could see how the spectra is changing with different levels of T-bars of oxidation. And this is the second overtone. This is another region. This is a second, and 740 is the third overtone. This is 960 is the second overtone of the water. Water and water. Now, this is what happened. These are the two controls. For 3 hours, when we have oxidation, the distance between the control and the oxidized sample is increasing. And here also, and here also. So we have a proof that we can diagnose the oxidative stress, and going back to the spectra, subtracted or analyzing multivariately, so we could understand which are the structures, which are the water matrix coordinates that are involved, that are making the biomarker of oxidation. And this is the spectral pattern. In mouse, when we analyze living systems like mouse, and for example, when we do human diagnosis monitoring, the individuality is very strong influence. So we all have our own signature, our own spectral pattern. On the top of it, so we have specific common bands that are activated with oxidation. And our recent study, very recent, just a little bit, we're working with probiotic bacteria, and we want to see what is the difference between probiotic, moderate, and non-probiotic lactic acid bacteria, just to show you how the aquagrams could show these differences. So this is probiotic. So we have the spectral pattern for probiotics. You could see this area that I just told you that this is a working horses kind of area, this hydrogen bonded area, and as well as dehydration band. And this is a part of, we don't have water shells here. No, we have water shells with probiotic, but not with moderate. And this is non-probiotic bacteria. Non-probiotic bacteria doesn't have hydrogen bonded structures, does not organize, and also do not have these free water molecules and dehydration band. So this is a language. So I would say we could use these water matrix coordinates as letters. So with these letters, we can write words. With these words, we could have a language, a water language that we could understand each other someday, probably. So we should be able to explain what is happening in water and how we could relate functionality of water to water structure, water molecular system, and other molecules. So, and these are just, if I subtract the spectra, this is what happens. This 1400 is a dehydration band. This is the water dimer, which seems to be very interesting. I know that it's interesting, but I'm not there. Not yet. It's very interesting place. And these are the hydrogen bonded part of molecular system. How many minutes do I have? Okay, I'm very close. I wanted to show you the absorbance of is around 0.36. And in here we have many spectra, plenty, but we don't see it. Now, these are different waters. And you see how many different functionality. This is 1452, which we call bulk water, and this is the dimer. Well, this is the bending vibration. This is the water shell. Look at the variations of the water shell. So every band means function. So by looking at this spectral pattern, we could think about functionality of each water and the differences. So this is how we can use. And for example, if we compare one particular water with molecule, with pure water, we can see where are the differences. And later, when we know everything about these bands, probably we will know what is the function of this new water. Okay, so I'm not going to, this is my lab, and I'm so grateful that I'm working with wonderful people, and I don't have all the collaborators here. Just we had recently a lab trip, but I'm really, really happy and blessed to be able to work with wonderful students and collaborators. And I use this to say thank you with my poem in Japanese and in English. Thank you.